When a sequencing machine reads a human genome, it doesn't produce one long, clean string of information. It generates hundreds of millions of short, overlapping reads, unstructured fragments scattered across a massive digital dump. These unorganized reads pile up into an astronomical file size, totaling over 100 gigabytes of raw data for a single person. If you try to open a 100 gigabyte file in a standard text editor, your computer will immediately freeze. Standard consumer software was never built to handle files of this magnitude. To solve this, scientists rely on a specialized pipeline of bioinformatics file formats. These formats are engineered to filter this data step by step, distilling the volume until the biological signal is clear enough to analyze. Mastering this specific digital pipeline is the only way for a researcher to sift through 100 gigabytes of noise and find the specific genetic variants that allow for a precise clinical diagnosis. The process starts with the FASTA format. It's the most basic foundation, containing just the name of a sequence and the raw DNA letters A, C, T, and G. But FASTA has a critical flaw. Every sequencing machine has a margin of error, and the FASTA format has no standardized way to store the confidence level of each base it identifies. This is why we use FASTQ. It pairs every nucleotide sequence with the machine's own accuracy estimate. This fourth line uses ASCII characters to encode the Phred quality score. Each symbol represents the mathematical probability that the base above it was called incorrectly. This chart tracks those quality scores across the entire length of a read. You can see the confidence levels are high at the beginning, but tend to degrade as the machine reaches the end of the sequence. By recording these errors, the FASTQ format allows researchers to programmatically discard low-quality data before it leads to a false positive or a misdiagnosis. Even with high-quality reads, the data is still just a collection of fragments. We don't know where these millions of reads actually sit within the 3 billion bases of the human reference genome. The Sequence Alignment Map, or SAM format, provides that context. It maps every raw read to its specific coordinates on a chromosome. The problem is that SAM files are text-based and incredibly bulky. They take up so much storage and memory that they become a bottleneck for downstream analysis. To fix this, we convert the text to BAM, a binary alignment map. It contains the exact same information, but in a compressed binary format that computers can process significantly faster. Further optimizations like the CRAM format save even more space by only recording the specific nucleotides that differ from the reference genome, ignoring everything that matches. Without this binary compression, the hardware requirements for large-scale genomic medicine would be too expensive and too slow to be practical. But a numerical coordinate isn't enough on its own. Knowing a mutation is at position 10,000 on chromosome 19 is useless unless we know if that position falls inside a protein-coding gene or a non-coding region. This is where annotation formats like GFF and GTF come in. They act as the master maps of the genome's landscape. These files define the boundaries of biological features, marking where one gene ends and the next begins, and identifying the specific exons and introns within them. By combining these formats, we turn a list of random coordinates into a navigable map of human biology. Most of a patient's DNA is identical to the human reference. If we are looking for the cause of a genetic disorder, 99.9% of the sequence is actually irrelevant information. The final step of the pipeline is the variant call format, or VCF. The VCF file ignores all the normal matching DNA. It records nothing except the specific mutations where the patient deviates from the reference genome. This represents the ultimate reduction. A process that started as 100 gigabytes of raw machine output is concentrated down into a single megabyte of actionable data. This 1 megabyte VCF file is the document a clinician actually uses to identify the mutation responsible for a patient's symptoms. Bioinformatics file formats are more than just digital containers. They are a sequential filtration system, turning 100 gigabytes of biological noise into a single life-saving signal.